The main objectives of this session are to revise the key concepts of cardiovascular physiology. We only have kind of an hour and a half, so we can't cover absolutely everything, but I think these are the kind of the core things that I want to try and cover today. So we're going to spend a while talking about the cardiac cycle, then we're going to talk about electrophysiology of the heart, cardiac muscle contraction, the control of cardiac function, and then we're going to talk a little bit about the different blood vessels. Okay, so we'll start with question 1. What part of the cardiac cycle is the A wave of the JVP associated with? Is it A, the bulge of the tricuspid valve with the ventricular contraction? Is it B, atrial relaxation? Is it C, atrial filling? Is it D, closure of the tricuspid valve? Or is it E, atrial emptying with opening of the tricuspid valve? Okay, so I'm going to put the poll up so everyone can try and vote. We'll leave it for about 30 seconds or so. Everyone can pop their answers in and then we'll have a look. Okie dokie, so we have majority of people are saying answer E, but then we've got a kind of equal split between A and D as well. A few answers for C as well, so let's have a look. Let's have a look at the answers. So the correct answer is closure of the tricuspid valve. Okay, so when we talk about the cardiac cycle, what we are often met with is this really scary picture, which is got a lot of lines on it and a lot of numbers, and it can be a little bit difficult to break it down. So I've broken it down into each of the different stages to try and help it make a bit more sense. Okay, so I'm going to start off by looking at the left ventricle. So here we have the pressures in the left ventricle. So this bottom axis here, the x-axis, that would be time, and this y-axis here, that would be pressure. So we're going from 20 millimeters of mercury up to about 120 millimeters of mercury, because we know a normal adult blood pressure would be about 120 systolic. Okay, we start off with the ventricle relaxed here, and it's just slowly filling here up until the point that the tricuspid valve will close and the ventricle starts to contract. So we have this really big upstroke here where the ventricle is contracting. At some point around here, the ventricular pressure overcomes the aortic pressure and we start to have the ejection phase, and then the ventricle slowly starts to relax, and that's this pressure reducing back down, and then we have kind of slow ventricular filling again. So the next part that we can really add in here, so now that we've got the left ventricular pressure, we can add in the aortic pressure. So the aortic waveform is probably the easiest one that we can think of, because we know what the systolic blood pressure usually is. It's about 120, and we know that the diastolic blood pressure is usually about 80. So this point here is the point at which the aortic valve opens and blood can start to flow from the ventricle into the aorta. And that is because the pressure in the ventricle is higher than the pressure in the aorta. So that would be the aortic valve opening and blood moving into the aorta. Obviously, because blood is moving into the aorta from a high pressure environment, the pressure within the aorta will start to increase. It will increase to just above the ventricular pressure, but because we've got such a momentum of the blood flow from the ventricle into the aortic valve remains open for a short amount of time, and blood continues to flow from the ventricle into the aorta up until this point here, and this point illustrated by this dotted line here. And that's at the point which the aortic valve closes and the blood flow ceases to flow from the ventricle into the aorta. This little notch here is the dicrotic notch, and that is to do with the elastic recoil of the aorta, and it kind of levels out the pressure within the aorta. Okay. The pressure within the aorta slowly decreases again to the point at which we've got our systolic blood pressure around 80, and then the cycle will repeat itself. So, because we've got a closure of the aortic valve, that's when the second heart sound is. Okay. Next, we're going to add in the JVP waveform. So that is the pressure in the jugular venous, or in the jugular vein. So, or just any kind of central vein, really. We start off with the pressure in the central venous system and in the atria being higher than that in the ventricle, and that is because blood is flowing from the atrium to the ventricle. We then have this kick of atrial contraction here, and then we have the tricuspid valve closes at this point here. At that point, the blood ceases to flow from the atrium into the ventricle. We have this C, the next bit here is this C wave here, and that is due to, because we've got the ventricle contracting here, we get a bulge up of the tricuspid valve up into the atrium. That's just to do with the contraction in the pressure of the ventricle. So we get an extra wave coming up here, and then that gradually starts to decrease. That is the X descent. Then we have an increase here, so another wave, so that's our V wave, and then we've got our last descent. Okay, so again, so we've got our sound 1, that's when our tricuspid valve closes, and then we've got our sound 2, that's when our atrial valve closes. This point here is when our tricuspid valve opens, and this one here is when our aortic valve opens. Okay. JVP doesn't look that good on this graph because all of the pressures are quite low. So I've drawn it out again. This is our JVP waveform, and I've drawn 2 repetitions of this just to make it clear that that's kind of a repeating thing that just keeps on going on and on. But to break it down into just the single bit, so we've got our A wave, so that's when our atria contracts. We've got our C wave, that's bulging of the tricuspid valve up into the atria. Our X descent, so that's as blood is flowing from the atria, or as blood is flowing from the ventricles to the atria, we get kind of a pulling of the tricuspid valve down into the atria, so it's kind of elongating the atrium, bringing that pressure down. We've got atrial filling here, which is our V wave, and then our Y descent here, that's when the blood starts to flow from the atrium to the ventricles again. Okay. So we start off with our A wave, then we've got our C wave, the X descent is this whole descent here, our V wave is this one here, and our Y descent is here. Okay. Question 2, which phase of the myocyte action potential is associated with the opening of voltage-gated calcium channels, and that allows calcium to flow into the cell while potassium flows out of the cell? Which phase of the myocyte action potential is associated with the opening of voltage-gated calcium channels? This allows calcium to flow into the cell while potassium flows out of the cell. Okay, so let's relaunch that poll. And if everyone can vote, so it's phase 0, phase 1, phase 2, phase 3, or phase 4. Okie dokie, so we've got the majority of answers are for answer C, and we've got a few for B, D, and A. So the answer is C. So well done, everyone. So here's our myocyte action potential. The myocyte action potential begins at minus 85 millivolts or thereabouts. That's our resting membrane potential. We get this sharp increase in membrane potential. That's due to our phase 0, where sodium flows into the myocyte. This is a really sharp increase, and that's just to do with the way in which the myocytes are connected. They have these gap junctions between them, which allows sodium to flow into them very, very quickly. So while some action potentials, they have a much shallower upstroke, this one's a very, very steep upstroke. Next, we have phase 1, and that's as potassium starts to flow out of the myocyte. Then we've got phase 2, our plateau phase. So that's when potassium is flowing out of the myocyte, but voltage-gated calcium channels open, and the calcium is flowing into the myocyte. And so we've got a net very slight downslope still, but it is kind of a plateau. And this is what stops the heart from being able to have tetany, which is where we have sustained muscle contraction. We can't have another action potential flowing because of this plateau phase, and that's kind of what protects our heart from having that. Phase 3 is as potassium starts to flow out of the cell, or continues to flow out of the cell, until we get to our resting membrane potential again, minus 85 millivolts. And then at phase 4 is our cell or myocyte is starting to return all of the ions back to normal via the sodium potassium ATPase pump. Okay, so next question. In pacemaker cells, which ion channel is responsible for the slow decay of resting membrane potential, which allows the cell to reach threshold potential? Is it A, voltage-gated sodium channels? Is it B, inward funny sodium channels? Is it C, L-type calcium channels? Is it D, T-type calcium channels? Or is it E, potassium channels? Okay, so I'll give you 30 seconds or so to answer. Okie dokie, so we've got about 52% of you all saying the inward funny sodium channels. We've got some answers for L-type calcium channels, voltage-gated sodium channels. Okay, so the correct answer is the inward funny sodium channels. So this question is talking about the slow decay of resting membrane potential. So our pacemaker action potential is slightly different to our myocyte action potential that we saw earlier. So we know that the heart generates its own electricity. It doesn't need nerves to initiate the cardiac action potential. We have our pacemaker cells to do that. These cells can do this because we don't have a flat line for the resting membrane potential. There's a steady decay from about minus 60 millivolts, which is where the resting membrane potential starts, to about 40 millivolts, which is threshold potential. This starts off from 60 millivolts to about 50 millivolts. That's just sodium coming into the cell via the inward funny sodium channels. After we get to 50 millivolts, the T-type or transient type calcium channels start to open. And so we have flow of ions via both of those channels. And that's up to about minus 40 millivolts. And that's when we reach threshold. That's when our L-type calcium channels open and we get a sharp increase in membrane potential up to plus 20 millivolts. At that point, these channels close and we have our potassium channels opening, our slower potassium channels, and they return our pacemaker cell membrane back to resting membrane potential, which is on minus 60 millivolts. Okay, so the next question. So, according to Frank Starling's law of the heart, which is the following is directly proportional to stroke volume in a healthy individual? Is it A, left ventricular end diastolic volume? Is it B, afterload? Is it C, systolic blood pressure? Is it D, heart rate? Or is it E, circulating volume? Okay, so let's have a look. So I've got the majority of people saying left ventricular end diastolic volume, a few answers for the others. So the correct answer for this one is left ventricular end diastolic volume. So the Starling curve, I'm sure that everyone will have seen this picture by now. It's essentially, Frank Starling did some experiments on frog hearts and determined his law of the heart. So what we know is that left ventricular end diastolic volume, as left ventricular end diastolic volume increases, so does stroke volume. This is kind of an exponential kind of increase of the curve. At this bit down here, so we have very small left ventricular end diastolic volume, which means that the sarcomeres are very close to each other and they don't have very much ability to move past each other and give a really big stroke volume. As the left ventricular end diastolic volume increases, we can still reduce our left ventricular end systolic volume to a similar amount, and therefore the stroke volume can increase. Having said that, there are a couple of lines that we need to think about. So this red line here is in a failing heart. So once we get past this point of the heart beating really well and ejecting, you know, a good volume of blood and having a big ejection fraction, that's when the sarcomeres start to become too stretched and the muscle is overstretched and it can't bring itself back to the volume that it was to begin with. And that's what we see here. So this is what we think about for the people that are more towards their clinical years. That's what we think about when we're giving too much fluid in someone that has a failing heart, and they're on this end of the curve, and their heart can't pump against that volume anymore, and they can't eject all of the blood that's in that, or a good proportion of the blood that's in that ventricle. On the contrast to that, so we've got this green line here, and this is a heart which has an increased ionotropy, which is an increased pumpability of the heart, essentially. It's just able to give that force of contraction a bit more than in the normal heart. So we have these different lines for the Frank-Starling curve. So we've got our failing, our normal heart, and then a heart which has got an increased ability to pump due to an increased ionotropy. Okay. Question 4. During cardiac muscle contraction, which component of the thin filaments binds the myosin heads? Is it A, troponin T? Is it B, tropomyosin? Is it C, troponin I? Is it D, troponin C? Or is it E, actin? So let's bring up our poll. Okay, so we just give another 30 seconds or so for you to have a think about this one. So I've got an overwhelming majority that are all saying acting, and I think that's because I clicked too far, and that explains the reason why of that. So apologies, everyone. I hope my explanation is still useful. So here is our sarcomere. We've got our thin filaments at the top. So we've got this green line, which is representing tropomyosin. We've got our little purple dots, which are representing actin. And then we've got these little blue dots down here, which are representing our troponin. So we've got troponin T, and that binds the troponin to the tropomyosin. We've got troponin I, which blocks the myosin heads from reaching the myosin binding sites on the actin. And we've got a troponin C, and that is the portion of the troponin which binds to calcium. We've got our thick filaments down here, and they're just made up of myosin, and there's multiple strands of myosin, all each with their myosin heads that bind onto the actin and help move the functional unit of the muscle along, because that's what the sarcomere is. Each sarcomere is a functional unit of the cardiac muscle. So here we are, so cardiac contraction. So this picture, so I've kind of done a bit of a series of pictures to try and, I'm a very visual learner, so it's just to try and help put it in everyone's brain of how this works. So I hope this works well for you as well. So we've got here our myosin head. We've just drawn one just for simplicity. And that myosin head has an ADP and an inorganic phosphate bound to its head. We've got our troponin unit here. We've got our tropomyosin and the actin. So this is the muscle during its resting state. When calcium is released into the muscle due to a action potential being generated and causing depolarization of the muscle, there's lots of calcium is released, and calcium binds to the troponin C. This causes a conformational change in the muscle subunits. So the tropomyosin and the troponin can move out of the way, and our myosin binding sites on the actin become free. At this point, the ADP and phosphate are released from the myosin head, and the myosin binds to this actin binding site, the myosin binding site on the actin. The myosin head has some stored energy in it, and this point here where the myosin moves, there's a conformational change in the myosin, is called the power stroke. So this pulls the thick filament forward along the thin filament. Next up, ATP binds to the myosin head, and this dislodges the myosin head from away from the thin filament. The myosin relaxes, and the ATP is broken down into ADP and phosphate, and the energy from that reaction is stored again in the myosin head. And that brings us to essentially the beginning. These calcium molecules will then dissociate, and it brings us to our relaxed state again. So here's another picture of a sarcomere, which again may or may not be familiar to you. So this is just a histological appearance of the muscle functional group. So we've got our M line, so that's the line in the middle where the myosin are all attached to each other, the thick filaments are all attached to each other. And we've got multiple different sections of thick filaments coming off of this M line. We have our A band, and our A band is the portion of the sarcomere which has the thick filaments in it. We have our I band, and that's the portion of the sarcomere which has only thin filaments in it. Our H zone is the zone which only has thick filaments in it, so it's not this portion where there's overlap is not included in the H zone. And then we've got the Z line, and that's the line which connects all of the thin filaments to each other. So as you can see, as the muscle contracts, the M line stays the same and the Z line stays the same, obviously. The A band stays the same in length, but the I band in the H zone will reduce in size because these thick filaments are being pulled across the thin filaments. Okay. So this is the same question as last time. We'll skip over this one. Question 6. What is the intrinsic rate of the sinoatrial node? Is it 30 beats per minute, 40 beats per minute, 70 beats per minute, 100 beats per minute, or 120 beats per minute? Okay, so the correct answer was D, which is what the majority of people have said. So we've got 40% are saying D and 30% are saying C. Okay, so although the sinoatrial node is generally classed as the pacemaker of the heart, we have various different pacemaker cells that are in the heart. So if the sinoatrial node was to fail, we have other bundles of pacemaker cells which can set a heart rate. So we have the sinoatrial node, which usually predominates, and that's because that's at the fastest rate. So by the time that the AV node decay, for example, was ready to initiate an action potential, there already would have been an action potential from the sinoatrial node, which would then reset the AV node. So we've got our SA node intrinsic rate is about 100 beats per minute, our AV node is about 40 to 60, and the bundle of His is around 50 15 to 40 beats per minute. Okay. So why is our resting heart rate usually about 70 beats per minute if the intrinsic rate of the sinoatrial node is 100 beats per minute? So that's due to the autonomic control of cardiac function. So the cardiac or the heart rate is set by the sinoatrial node, but we also have sympathetic and parasympathetic innervation to the heart, which can upregulate or downregulate the heart rate. And at rest, it's the parasympathetic which predominates. These two are the wrong way round. So let me just alter that. So here we are. So predominantly the parasympathetic innervation predominates at rest. So that brings the heart rate down. So if we remember what sympathetic and parasympathetic mean. So parasympathetic is our rest and digest, whereas sympathetic is our fight and flight. So if we think about it, sympathetic, we want our heart rate to beat really fast and we want a really good blood pressure because we want to run away. Whereas the parasympathetic, we're kind of like just sitting in our sofa after having our Sunday dinner, kind of relaxing, okay? So the parasympathetic innervation is via the vagus nerve. So that's what this green line is trying to demonstrate, whereas the sympathetic input is via the sympathetic chain. Things that feed into those inputs to help regulate heart rate. So we've got our high pressure baroreceptors. So they're the aortic and carotid sinuses, and they are found in the internal carotid artery and the aortic arch. They sense high blood pressure, and when the blood pressure is high, they fire very, very quickly. They're firing all the time, but when blood pressure is low, they're firing very slowly, and as blood pressure increases, they fire quicker and quicker. We also have our low pressure baroreceptors, which will fire based on blood pressure being low, and they are found in the heart and also in the coronary arteries. So here's a little schematic of the autonomic control of cardiac function. So this is kind of looking into the types of receptors and the type of nerves that feed into the heart. So our parasympathetic at the top here, so this is our vagus nerve, parasympathetic. So we've got our nicotinic acetylcholine receptors and then our muscarinic acetylcholine receptors, and they reduce heart rate, reduce vascular tone and keep blood pressure low. And then we've got our sympathetic here. So that would be the things that increase our heart rate. So we've got our fibers from the sympathetic chain, which go directly to the heart. We've got our noradrenaline receptors. Our adrenoreceptors can be alpha receptor adrenoreceptors and beta adrenoreceptors. The alpha adrenoreceptors are predominantly in the blood vessels and they cause vasoconstriction, whereas our beta adrenoreceptors are predominantly in the heart and they increase heart rate. The way that because I used to get really confused about this, and the way that I now remember which ones are alpha and which ones are beta adrenoreceptors are, if you think about beta blockers, they're blocking your beta receptors and slowing heart rate. Therefore, the beta receptors, when they are activated, must increase heart rate. And that's how I remember it now. In the sympathetic nervous system, we also have nicotinic acetylcholine receptors giving off a postsynaptic neuron to the sweat glands, and then muscarinic acetylcholine receptors. And then we've got our dopamine D1 receptors, which again cause an increase in heart rate and a reduction in vascular tone this time. The last portion of the sympathetic nervous system is the nervous supply to the adrenal medulla. So that again, because it's a So something to mention, so if you notice, all of our presynaptic neurons will synapse onto nicotinic acetylcholine receptors. So our adrenal medulla is, although it's an kind of the end organ, it is part of our presynaptic neuron is then synapsing onto the adrenal medulla. The adrenal medulla releases adrenaline and noradrenaline directly into the bloodstream, and the adrenaline and noradrenaline again will work on these alpha and beta receptors, so they'll cause vasoconstriction via the alpha receptors and an increase in heart rate via the beta receptors. So that's kind of how the autonomic control of cardiac function works. So we've got a variety of different receptors and a variety of different neurotransmitters, our adrenaline and noradrenaline, our dopamine, and our acetylcholine in the sympathetic nervous system. And then we've only got acetylcholine as the neurotransmitter for the parasympathetic nervous system. In the parasympathetic nervous system, that's the one with the vagus nerve, we've got a very long presynaptic neuron and a very short postsynaptic neuron. Whereas in the sympathetic portion, we've got a very short presynaptic neuron and a very long postsynaptic neuron. And that's the only difference of that is in the adrenal medulla, where all we have is a presynaptic neuron. And then after that, it's the bloodstream which carries the neurotransmitters. Okay. Question 7. An increase in which of the following hormones causes a reduction in circulating volume and cardiac output? Is it A, dopamine, B, atrial natriuretic peptide, C, adrenaline, D, aldosterone, or E, thyroxine? So bring up the poll again. Okay, so around 70% have said atrial natriuretic peptide or ANP, and around 20% aldosterone, a few for thyroxine. Okay, so the correct answer was atrial natriuretic peptide or ANP. So there are quite a lot of hormones involved in the control of cardiac output. So I've broken them down by system. So we've got our adrenal medulla hormones, the adrenaline and noradrenaline. We've already discussed those. We've got our hypothalamic hormones, dopamine. Again, we've discussed that previously, so I won't go into any more detail on those. We have hormones from the posterior pituitary gland, so that's a vasopressin or antidiuretic hormone. We've got our thyroid hormones, T3 and T4. We've got our renin, angiotensin, aldosterone pathway. So we've got renin, angiotensin 2 and aldosterone. And then we've got our cardiac hormones, atrial natriuretic peptide and brain natriuretic peptide. So that's your ANP and BNP. All of these top ones are involved in increasing blood pressure, some of them increasing heart rate as well. Whereas the cardiac hormones, these two at the bottom, they're involved in reducing blood pressure and reducing cardiac output. Okay, so we'll start with the pituitary. So the pituitary releases vasopressin. It's stimulated by a number of things. So hyperosmolarity is an increase in number of osmotically active particles in the blood. So it's very closely linked with hypovolemia. we've also got hypotension, so low blood pressure, and then we've got our sympathetic stimulation and angiotensin II. We're going to talk about the angiotensin II later, so don't worry about that one for now. Vasopressin can act via two different receptors. So we've got our V1 receptors, which cause direct vasoconstriction, and that in itself causes an increase in arterial blood pressure. We've also got our V2 receptors. They're involved in releasing aquaporins in the collecting duct of the kidneys, and that causes increased water retention from the kidney, causes an increased circulating volume, and in that way it also increases blood pressure. So we can have direct vasoconstriction and also increasing circulating volume. Next up, we've got our renin-angiotensin-aldosterone system. I used to hate learning about this as a medical student, but I promise you it doesn't go away. You still have to learn it now. I still had to learn it for my exams I've just done, so it's worth just getting your head around it. Angiotensinogen is our precursor protein, which is broken down into angiotensin 1 when renin is released from the kidneys. Renin is released in response to a low blood pressure. Angiotensin 1 is then cleaved by angiotensin converting enzyme in the lungs, and it becomes angiotensin 2. Angiotensin 2 then can work in a variety of ways. So angiotensin 2 itself can cause systemic vasoconstriction, and that will increase blood pressure. As we said before, it can cause release of vasopressin. And angiotensin 2 also can cause aldosterone release from the adrenal cortex, and aldosterone causes sodium and water retention. And that's generally in the distal convoluted tubule of the glomerulus. ANP and BNP are released in response to stretch. So when the heart is overstretched because it's too full, ANP and BNP are released. What it does is they work in two ways. So it can increase renal filtration, and that's by increasing the blood flow to the glomerulus and allowing more solute to be filtered, and therefore more water is excreted by the kidneys. We also get a reduction in systemic vascular resistance, so the heart has less to pump against, and therefore it can empty itself better. BNP, for those who are in your clinical years, BNP is one of our markers that we measure for heart failure. So when the heart is overstretched and failing and it can't pump effectively, lots of BNP is released, and that's kind of where that relates in a clinical setting. But for your preclinical years, it's just important to know that they are involved in a reduction in cardiac function and to try and reduce the volume, trying to reduce what the heart is pumping against, just to try and keep the system working. Next, which of the following factors causes coronary artery vasodilatation? Is it increased aortic pressure, increased heart rate, increased adenosine, atheroma, or hyperoxia? We'll bring up our poll again. Okie dokie, so again, we've got 54% saying increased adenosine, And then we've got a few for increased aortic pressure, increased heart rate. Okay, so the correct answer is increased adenosine. So, a few things about coronary artery blood flow. So, blood can only flow in the coronary arteries when the blood pressure exceeds the pressure within the cardiac muscle. Okay, so what I've tried to demonstrate here, so during systole in the left ventricle, as you can remember from when we were looking at the cardiac cycle, our systolic blood pressure will be about 120 millimeters of mercury, but so will the pressure within the left ventricle itself and the pressure within that muscle. And therefore, during systole, so during when the heart muscle is contracting, there will be no flow in the left ventricle, so blood can't get to the left ventricle. During diastole, though, we've got a diastolic blood pressure of about 80, and our pressure within the left ventricle during diastole is somewhere around 5-10 millimeters of mercury, not very high. So we will have flow. There will be a 75 millimeter of mercury perfusion pressure. And so what I'm trying to demonstrate from this slide is, particularly in the left ventricle, which is the high pressure portion of the heart, we only have blood flow during diastole. And why is this important? So in tachycardia, so when the heart's beating really fast, there's a reduction in the amount of time that the heart is in systole, and also the amount of time that the heart is in diastole, because obviously everything is happening quicker. So for each cycle of the cardiac cycle, it's taking place over a short amount of time. However, it's disproportionate. The reduction in the time for each of those is disproportionate. So we have a longer proportion of the time is spent in systole, so spent contracting, than spent in diastole, so relaxing. And once we get up to heart rates of kind of 150, things like that, the amount of time that we're spending in diastole, where the coronary arteries can fill and we can deliver oxygen to the left ventricle, is so minimal that we're really struggling to perfuse that ventricle. We're struggling to be able to deliver oxygen and nutrients, and we're struggling to be able to take all of those waste products away. So that's quite important of why we don't like patients to have very high heart rates. As well as the increased demand, there's also a reduced blood flow to that left ventricle. Okay, so another thing that we can talk about when we talk about coronary blood flow. So we can talk about the heart as having an autoregulation system. What that means is that over pressures of between about 40 to 160 millimeters of mercury, the flow through the coronary arteries remains very similar. So we can still get blood flow to the heart quite well between those two values. Mechanisms of autoregulation. So when we need increased blood flow to the heart muscle and we need the vessels to relax, the vascular endothelium, via various different mechanisms, releases more nitric oxide, and that helps to dilate, or that works on the smooth muscle of the coronary arteries, that causes vasodilatation, allowing more blood flow to easily get through those vessels and to be able to perfuse that myocardial muscle. Another thing which would make a lot of sense, and which is why we've adapted to have this, so local metabolites will increase vasodilatation. What this means is an increase in adenosine, which is a breakdown product of ATP being broken down into ADP, etc. We if we've got increased amounts of adenosine, that again will cause vasodilatation. Other things would include things like potassium and those kinds of breakdown products when we're undergoing things like anaerobic respiration and things like that, and all of those metabolites are starting to build. That will encourage vasodilatation and encourage blood flow to the heart. And then we've got neural factors, so things that we've spoken about in terms of all of the nervous system involvement in getting the heart pumping are also involved in getting blood flow to the heart muscle. Question 9. Which type of blood vessels provide the majority of the systemic vascular resistance? So, is it A, arteries, B, arterioles, C, capillaries, D, venules, or E, veins? Lovely stuff. Okay, so the majority of people have said B, which I agree with. So we've made some pictures of the different types of blood vessels. So our arteries and veins both contain the same layers within the blood vessels. We've got our external layer, which is the tunica adventitia. We've got our tunica media and our tunica interna within both types of those vessels. Tunica adventitia contains a lot of fibrous tissue, which basically stops the vessel from popping. And our tunica media contains a lot of the muscle layers, which help to change the tone of the vessel. You'll notice that the arteries have much thicker layers of these. That's because the arteries are a much higher pressure system. So they need to be able to regulate the blood pressure to stop huge swings in blood pressure, which we don't really want, and also to be able to control where blood is going. Arterioles don't have this external layer. They just have the tunica media, which contains a couple of muscle layers, and then we've got our interna, which is kind of the endothelial layer. Capillaries here are one cell thick vessels, and they're ones responsible for gas exchange. Our venules, they don't really have very much muscle layer. They do have the fibrous layer. And then our veins are kind of our big floppy things that don't really contain that much muscle. They can't really contract and relax as much as our arterioles and our arteries do. The main reason why arterioles provide more systemic vascular resistance is we've just got so many of them, and they can really, changes in arteriolar diameter can cause big differences in our systemic vascular resistance, whereas arteries, we don't have quite as many of them, or they don't contain quite as much volume of blood. Our veins are what we consider to be capacitance vessels. So what that means is the veins will contain about 60 to 65% of the blood in our body at any one time, whereas arteries will only contain about 15%, something like that. Question 10, where in the body are fenestrated capillaries found? Is it the liver, the brain, the heart, the lung, or the kidney? Okay, lovely. So 62% of you are saying the kidney, and I agree. So we've got a few different types of capillaries. So we've got our continuous capillaries. So these are where we've got a cell and then another cell and then another cell straight away. We've got our fenestrated capillaries, where they've got little holes within the endothelial membrane, which allow larger particles through. And then we've got our discontinuous capillaries. That's where we've got kind of big holes in our capillaries. The fenestrated capillaries will be found in places like our endocrine glands, the kidney, and the intestinal villi, and that's when we want to allow slightly larger particles through than what could fit through our continuous capillaries. And our discontinuous capillaries are where we want kind of cells to be able to fit through. So that's our liver, our spleen, and our bone marrow, where kind of red blood cells, white blood cells, things like that are able to fit through those holes and get into our bloodstream. We've also included here sinusoids, which are found in the liver, which can sometimes be considered a type of capillary to an extent, but I won't go into massive amounts of detail of those because that would be more liver physiology, so I don't want to get bogged down in that. When we think about capillaries and we think about diffusion of water through the capillary, we think about something called Starling's forces, which I've tried to illustrate here. So this red section here is our inside our capillary, that's blood. And this purple section here, that's outside of our capillary, so that's our interstitial fluid. We've got our capillaries, our endothelial cells here. Within the capillaries, we've got large proteins which can't fit through the capillary membrane. And so while some certain things, so things like carbon dioxide, non-polar small molecules would be able to just diffuse through the capillary membrane. Then we might have some channels which let other solutes through the capillary membrane. These large proteins can't fit through at all unless there's damage to those capillaries. So we have a few different forces that come into play when we think about diffusion of water through a capillary. So first of all, we need to think about the pressure within the capillary. So I've labelled here the arteriolar end and the venular end. So that's the side where it's next to the arteriole. This is the side where we're next to the venule. At the end where we're close to the arteriole, we've got a capillary pressure of somewhere around 35 millimeters of mercury. So that force is a force within the capillary, which is pushing fluid from the capillary out into the interstitial fluid. We also can consider, but I haven't included it on this diagram because it would be negligible, the pressure from the interstitial fluid into the capillary. But it's not worth considering it because that, like I say, would be negligible. There isn't really a pressure pushing the interstitial fluid into the capillary. The next thing we need to consider is the oncotic pressure, or as we will have learned from our A-levels, we'd probably call it osmotic pressure. So this is the pressure that these large proteins, because of the large proteins are solutes within the blood, they are increasing the osmotic or oncotic pressure of the blood. So we've got an oncotic pressure here of about 28, and that's pulling the water from the interstitial fluid back into the capillary. And then we've also got an oncotic fluid of the interstitial fluid, but because that doesn't really contain big proteins, that's only about 3 millimeters of mercury. So our net filtration is 35 minus 28 minus 3, so that's our pressure, our blood pressure essentially pushing out, minus the oncotic pressure, what's pulling fluid back in. And that's about 10 millimeters of mercury out of the capillary at the arteriolar end. As we move along the capillary, the pressure within the capillary reduces. That's because all of this fluid is getting pushed out. And also we don't have very much to work against. So the veins, as we've discussed before, are very low pressure systems. So the pressure at the venule is probably somewhere around 15 millimeters of mercury. The oncotic pressure remains about the same. And so our net filtration at the end of the capillary is about 10 millimeters of mercury in. And this is why we don't get a net loss of fluid constantly into our interstitial fluid. And we know that because we're not all really boggy and edematous and kind of essentially just filled with water. We're able to bring all of that water that we push out into our interstitial fluid back into our capillary. So that is the end of our discussion today. So we've covered the cardiac cycle, we've gone over the electrophysiology of the heart, we've spoken about cardiac muscle contraction, control of cardiac function, and we've spoken about the different blood vessels. So at this point, thank you very much everyone for listening. At this point, I'm going to try and open up the chat if I can. I'm sure it's possible. And I'll try and answer some questions. So.
Thanks so much, Hannah. That was really, really good. Yeah, I've been sort of following the chat. I think there were a few questions. There's people who had some questions. You just ask them again. And yeah, go ahead, sorry. And thank you so much, by the way, to Brighton Sussex Cardiothoracic Society. Sorry, I didn't say in the beginning, I completely forgot, for helping us to organize. It's been really, really helpful. Thank you. Yeah, sorry.
So we've got, can we go over the receptors again? So yeah, so we can. So let me go back to my slide. So here we are. So these are our receptors for the autonomic control of cardiac function. I'm just going to edit this because I've written these wrong again, and I don't want to, if anyone's writing this down, I don't want it to be incorrect. So here we are. So this is our autonomic control of cardiac function. So we've got our parasympathetic input, which will slow the heart rate. Because again, if we think about it, so parasympathetic is rest and digest. So we want to have our heart rate to be slow. We want to be able to relax, be able to, you know, rest and digest. So we can't have that really fast heart rate that's going to just make us feel really sick. So our parasympathetic is the vagus nerve, as we discussed before. That's our cranial nerve 10, and that comes from the brainstem. It's a really long nerve. We have our, so that's our preganglionic nerve, and it synapses quite close to the heart and to the blood vessels to our via nicotinic acetylcholine receptors. And then we have very small postganglionic fibers, which will synapse to the heart and to the blood vessels. And that will be via muscarinic acetylcholine receptors. So in the parasympathetic control of cardiac function, the receptors are always acetylcholine, but to begin with, we've got nicotinic acetylcholine receptors, and then closer to the heart, we've got muscarinic acetylcholine receptors. The sympathetic control is slightly more complicated in that we've got a few different types of neurotransmitters involved. So, but there are some similarities between the sympathetic. So all of the sympathetic nerve fibers, the preganglionic nerve fibers, are all short. And they all synapse via nicotinic acetylcholine receptors to their postganglionic nerves. At the top here, we've got our postganglionic nerves, which innervate the heart and the blood vessels via noradrenaline as the neuroreceptor. And we've got our beta adrenoreceptors, which will be on the heart, and that causes the heart rate to increase. And as we said before, we can remember this because beta blockers slow the heart down. And therefore, if those beta receptors weren't being blocked, they would speed the heart up. So our beta receptors are the ones that are to do with increasing heart rate. And our other receptors are the alpha receptors, and they're the ones that cause vasoconstriction and increase the blood pressure that way. To the sweat glands, we've got muscarinic acetylcholine receptors, so that's similar to the parasympathetic, but obviously the nerves are the opposite way around. And then we've also got dopamine D1 receptors, which innervate the heart, and they cause an increase in heart rate, but they also cause vasodilatation. For the adrenal medulla, that is innervated differently to all of the other organs. And so the adrenal medulla is innervated by preganglionic nerves, and they are nicotinic acetylcholine receptors. The adrenal medulla releases adrenaline and noradrenaline, which travel via the blood to the heart and the blood vessels, and that will cause an increase in heart rate and vasoconstriction. Both of those increase blood pressure. Okay. What is vasopressin, and how many cells in the heart are pacemaker cells? Do they all stimulate at the same time, pacemaker and non-pacemaker? So vasopressin is a hormone, which is also called antidiuretic hormone. So it's a little bit confusing, and these names get used interchangeably, and we just are supposed to just know that they're the same thing, and no one necessarily tells you that, but they are the exact same thing. Vasopressin can both cause vasoconstriction, so it's a drug that we use to cause vasoconstriction on the ITU when adrenaline and noradrenaline haven't worked. But it also causes increased water retention. So in the kidney, the last step of urine being produced from the kidney is the filtrate going into the collecting ducts. Vasopressin can cause proteins called aquaporins to appear on the collecting duct membrane. And that's via our V2 vasopressin receptors. And that will allow water to move from the collecting duct back into the bloodstream. And that increases circulating volume and causes an increase in arterial blood pressure. Does anyone have any other questions? Or is everyone happy?
Please fill in the feedback, guys.
Okay, lovely.
Oh yes, Shaylee. Do you want to tell us about your conference?
Hi, I'm Momina. I'm the events manager for CTSS. I just wanted to let you guys know that we have a conference running on the 1st of May. It's the International Cardiothoracic Society Conference. We're going to be holding some very interesting lectures and some workshops as well. Tickets are still open for sale, so if anyone wants to get a ticket, you can follow the link. And if you have any questions about it, you can go to our Facebook page or our Instagram page for more information. Thank you.
We'll put it up on our Quesmed's page, guys. Send me the details, please, and we'll put it up.
Sure, no problem. Thank you.
Thanks. Good. Right. Thanks, guys. I think, Hannah, anything else you wanted to add?
I don't think so. I think if there's no more questions, then I think we've kind of gone over the main things that I was hoping to cover today.
Brilliant. Good stuff. Good stuff. Cool. Grant, so I'll put up the recording, guys, in maybe a day or so. And yeah, sorry to say a million times, but please fill in the feedback. It's very useful for everyone. And yeah, that's it.
Right. Thank you very much for listening, everyone. See you later.
Thanks all. Take care. Thank you, Hannah.